Okay, dear student, last step is there, that is translation. So in the translation, what happened? The synthesis of polypeptide chain by addition of amino acid. There is 20 amino acid. So there is a all the RNA, mRNA and rRNA and tRNA, the three are play important role in the process of translation. How? In the translation, that is mRNA, that is 5' UTR and 3' UTR untranslated region before the initiation codon is present and after the termination codon. So this mRNA having coding sequences in the triplet form and act as a substrate for the binding of ribosome. This mRNA. mRNA, this is messenger RNA, carry the codons, triplet codon, where the tRNA comes and bind. So it acts as a substrate also for the binding of ribosome. Ribosomal RNA act as a protein factory, catalyze the reaction and play role in the elongation by moving from one triplet to another triplet. And transfer RNA, adapter RNA, tRNA carry different 20 amino acids and they require 20 different tRNA and making the polypeptide chain. Clear beta? When the initiation complex forms, mRNA, when ribosome small subunit comes and bind to the initiation codon, that form the initiation complex. Then the large subunit of the ribosome comes and bind to the mRNA chain. This large subunit having two sides, that is P and A site. When the two tRNA comes close proximity, they form the polypeptide chain or dipeptide chain. Peptide bond formation. So first tRNA which initiated tRNA and initiate the process, first tRNA, that is called methionine tRNA, which bind to the initiation codon and called methionine tRNA and is come to bind to the P site. That initiation is start now. Now subsequently, continuously, the new tRNA, charged tRNA molecule comes. Amino acid plus tRNA, that is called charged tRNA, amino acid transferase enzyme with the help of, and the charged tRNA comes and bind to the new tRNA to the A site, and both the tRNA, the close proximity P and A, they form peptide bond. Now the ribosomal unit which help in the peptide bond formation and catalyze the reaction called ribozyme activity, that is 23S RNA, very important, which catalyze the reaction. Now this ribosome move from next triplet, move to the next triplet, and this uncharged tRNA exit from the E site. Now the next tRNA comes to the A site and bind, and next chain is formed. Continuously the 20 tRNA comes to the A site and making a 20 amino acid and making polypeptide chain by moving the ribosome. When the ribosome reaches to the termination codon UAA and it receives the signal by termination, that start the process and growing RNA polypeptide chain release out. That process is called translation. Clear about beta? So these are the process of molecular, and I hope that you can understand all the things neat and clean. Okay, best of luck.